Belleperche. I'm back, and being about halfway through my PhD, I'd like to show you the Aptamer switch that I've spent the last couple of years working on. So I'm sure that being here, you're all familiar with oxidative stress as a concept. A lot of you probably work with it in one way or another, and some of you have likely published one or more of the 26,000 papers in 2021 indexed in PubMed on the topic. And there's definitely a lot to cover, since while reactive oxygen species are created and destroyed as part of normal metabolism, the dysregulation in ROS levels that we call oxidative stress is associated with many different disease states, as well as, generally speaking, the DNA damage caused by reactive oxygen species. Which is why tools for measuring reactive oxygen species and oxidative stress within the cell are a fairly intense area of study right now. But one of the issues with many of the current tools is that either they can only detect one specific form of reactive oxygen species, or they can't be used to monitor changes over time within the living cell. And this is in part because of the constant destruction of reactive oxygen species within the cell by natural antioxidant pathways, meaning that they have a very short half-life. So when we began to outline this project, we chose to focus on, instead of ROS themselves, a more stable product of oxidation. Of course, I mentioned DNA damage, and oxidation of the guanine base is by far the most common form of oxidative damage to the DNA, meaning that 8-oxidized deoxyguanosine has been used as a marker of DNA damage in the past and as an excreted biomarker. But of course, guanine isn't only present in the DNA or RNA, and the GTP pool within the cell is at concentrations of millimolars. And not only that, GTP is as much or more susceptible to oxidation as guanine is within DNA or RNA. So because of this, we've chosen to focus on oxidized GTP as a potential biomarker of oxidative stress within the cell. That as the GTP pool as more GTP is created and damaged GTP is removed, can change over time, making this a steady monitoring system. Now, of course, you all know what aptamers are, so I'll just say that the advantages of using an aptamer for this purpose are the reversible flexibility of the DNA and RNA structures, meaning that we can focus on making a fused biosensor switch, and the fact that RNA aptamers can be expressed stably within a cell line, meaning that we can have a self-reporting, non-invasive biosensor. Now, fortunately for us, to start this project, we were able to use a previously selected RNA aptamer for 8-oxodeoxyguanosine by Rink et al. In 1998. The aptamer was selected using 8-oxodeoxyguanosine on a column, but counter-selected using non-oxidized deoxyguanosine. But once the selection was complete, affinity testing was done with strands of DNA containing oxidized guanosine. And as you can see, from the EMSA assay, they found what they approximated to be nanomolar affinity for oxidized deoxyguanosine, while no detectable affinity for guanosine without oxidation. Not only that, by changing the position of the oxidized guanosine residue, they confirmed that it could bind in multiple positions within the DNA strand, suggesting that for our purposes, this suggested that it might be able to bind to other guanine-containing species. Our first goal with this project was to confirm that it could bind to free oxidized deoxyguanosine in solution. And for that purpose, we ordered the aptamer and sent it off to Bind in Germany, where they performed microscale thermophoresis testing. This confirmed that the aptamer was able to bind to the free small molecule in solution, but also gave us a KD in the high nanomolar range, 813, which was a little bit high for our purposes, but we weren't done with the aptamer yet. We wanted to fuse this aptamer into a biosensor, an RNA switch. So for that purpose, we wanted to make the aptamer smaller and therefore easier to work with. And fortunately for us, we found that the second stem on the aptamer is entirely unnecessary for binding. Not only that, by minimizing the aptamer, we improved the KD by over an order of magnitude. And this meant that it became perfect for our purposes because from previous mass spec measurements done, the estimated concentration of oxidized GTP within the cell is 20 to 90 nanomolar, which means that the KD we have of this minimer is perfectly within that range and extremely suitable for our intended purpose. Further mutation experiments also told us something interesting about this particular aptamer structure. Predicted structure for the aptamer actually varied between a simple stem loop and a smaller loop with a bulge. And we found that any mutation to the aptamer that favored or froze it in one of those two conformations reduced or eliminated binding, suggesting strongly that the top of that stem is highly involved in target binding, and that something interesting is happening with the binding structure. Now, of course, we needed to confirm the specificity of this aptamer as well, especially in the minimer state, and we had a lot of mutation analyses to do. So for these experiments, we used an assay that my professor previously developed for highly sensitive small molecule aptamer kinetics. And this is a modification to standard SPR, where instead of modifying the chip directly with the aptamer, the chip is functionalized with a long string of poly-T DNA. The aptamer is synthesized with a poly-A tail attached to either the 5' or 3' end. The small molecule target is flowed through as normal so that binding can be detected. And after each cycle, the surface is regenerated, washing off all of the aptamer and thereby both preventing residual signal from small molecule remaining bound to the aptamer and preventing the RNA aptamer from being damaged over multiple cycles. We were able to confirm that the minimer we created had retained its high affinity for not just 8-oxo-deoxyguanosine, but other oxidized guanine species, including 8-oxoguanosine and 8-oxo-GTP, our intended target. Not only that, we detected no binding to any non-oxidized guanine species, again, including GTP. And this is extremely good for our intended purpose, as of course, there's about 100,000-fold difference in concentration between GTP and oxidized GTP within the cell. Now, for our eventual purpose, we'd like to have this biosensor working not just within single cells, but within a stable zebrafish line. Inside a zebrafish embryo, if we can have a stable expressed RNA biosensor, we could both quantify oxidative stress and localize it within different tissues or different areas of individual cells. Zebrafish embryos are commonly used right now to study development, and it's known that reactive oxygen species play a part in several developmental pathways. Not only that, zebrafish embryos are being increasingly used to study the potentially damaging effects of various pharmaceuticals, and being able to localize oxidative stress to various tissues or even different areas within the cell as a self-reporting system could be extremely useful for these purposes. An endogenously expressed, minimally invasive probe would be ideal for this, particularly as it can be used on the live cells over time. Now, of course, to actually create this probe, this biosensor, we need to fuse our initial aptamer with something that creates a signal. And for this, we're using the broccoli aptamer, one of several fluorescent aptamers developed in the last few years. The broccoli aptamer binds to DFHBI, or one of its descendants, including DFHBI1T, which we are using. And by restricting the motion of the dye, causes it to fluoresce. In the absence of the aptamer, the dye has a very low background signal. By fusing our aptamer to the broccoli aptamer through a short transducer, we destabilize the broccoli aptamer. This creates a turn-on logic-gated sensor, so that only in the presence of our target, 8-oxo-GTP, is the full aptamer stabilized, allowing broccoli to bind its dye and create a fluorescent signal. In addition, the full switch contains a third tRNA or three-way junction or other stabilizing element to help it retain its structure within and fold correctly within a biological environment. And recently, we've been able to create a functioning switch. The graph on the left shows three separate conditions. Furthest to the left in green is a negative control condition containing just 8-oxo-DG, a random RNA sequence, and the dye. As you can see, there is a low background signal from the dye that is increased in combination with broccoli. The central condition in orange shows the aptamer with the dye alone. We do have some constant binding of the dye to the aptamer, as the broccoli is not completely destabilized in this design. However, in the third condition in blue, I show the effects of 8-oxo-deoxyguanosine over the course of an hour, where by binding to the aptamer, stabilizing the aptamer, we see a distinct increase in the fluorescent signal. This was one of 8 switch designs that I tested, and on the right you can see a non-successful switch design where after the course of an hour there is no increase in signal when 8-oxo-DG binds, suggesting that either the broccoli aptamer wasn't destabilized enough, or that it's not stabilized sufficiently by 8-oxo-DG binding, or that 8-oxo-DG wasn't binding at all. Now, obviously, there's a long way to go with this project. So what I'm working on right now is expressing this aptamer within the in vivo environment to confirm that it can actually work for our purposes. I'm starting off with E. coli for ease of expression through a plasmid vector. Once I've confirmed that I'm able to express this aptamer and it folds correctly in vivo, I'll be working on optimizing the structure. The three goals for this project are brightness of the aptamer sensor itself, speed of reaction, and of course sensitivity, how much of a change in oxidative stress level I'm actually able to detect. I expect this to take a while, but once I have that final best aptamer sensor, we'll be working on expressing it in mammalian cells as a potential alternative reporter mechanism, and finally creating our zebrafish and creating our stable zebrafish line with self-reporting oxidative stress detecting embryos. I'm really looking forward to what we'll be able to produce with this, but right now I'd really like to thank my university, and particularly the chemistry department, for all of the help and support I've gotten, my various sources of funding, NSERC, CRBS, FRQNT, TwoBind for the incredibly useful data we got with you guys, and the entire McKeag lab, including especially my professor and mentor, Maureen McKeag. Thank you, and I'm happy to take your questions.